Hello everyone, welcome to this webinar as part of our Shimadzu Solution for Pharma series. I'm Brian Demansky, the Strategic Collaboration Scientist at Shimadzu Scientific Instruments. I'll serve as the moderator today, and we will hear from Stephen Swartz about chiral chromatography and separation method development. Before we start, I wanted to share a couple of notes for our viewers. The webinar console has a variety of items to help enhance your experience and interaction with us. In the screen, you'll see the following items. The slides will appear in the center of the console. Directly under the slides, you will see the related content list with clickable links relevant to the material being presented today. On the top left of the slides is the Ask Question widget. Please submit your questions during the presentation through this widget, and we will answer them during the Q&A session. Just below the Q&A box are survey questions that you may fill out anytime during or after the presentation. On the right are the moderator and speaker bios. You may expand the items here to learn more about us. Finally, at the bottom of the pane are the icons to bring up all of these widgets in case they are minimized or hidden during the presentation. All right, let's get started with today's presentation. If you're just joining us, I'm Brian, your moderator, and today we'll hear from Stephen Swartz, and our topic is chiral chromatography and method development in pharmaceutical research. Stephen currently works as the Eastern U.S. Business Development Specialist for Pharma and Biopharma at Shimadzu Scientific Instruments and has over 30 years of experience in the pharmaceutical industry, including medicinal chemistry and early discovery drug work, as well as extensive knowledge regarding separations and purification. With that, I'll hand it over to you, Steve.
Thank you, Brian, for that introduction, and thank you all for joining us today. I hope you will enjoy our webinar. So let's get started. Here's a quick outline of what we're going to go over today. First, we'll start off with some basics in chiral molecules, what they are, and why they're important in the pharmaceutical industry. We'll then get into the basics of chiral chromatography, talk about molecular recognition and how this impacts chiral stationary phases. We'll then get into some method development, basics of screening and developing a screening platform, and then talk a little bit about instrumentation. Okay, so what does it mean when we're talking about chiral molecules? Well, chiral is a Greek term meaning handedness. So much like your hands are somewhat identical, they're not superimposable, they're mirror images. Molecules have that same kind of characteristics. They typically have a single chiral center or stereocenter. Usually this is a carbon atom, but it can be a nitrogen, phosphorus, or sulfur atom. One simple chiral center develops two enantiomers. More than one develops diastereomers, or sets of enantiomers. So in this slide, I want to show a couple different types of chiral molecules. The ones that we're most familiar with are small molecules that we see in pharma, ag, and food. The two examples I show here are ibuprofen with a single stereocenter and remdesivir, which has multiple stereocenters. Chiral molecules can also be planar. These molecules typically have some sort of structural constraint that holds them into a spatial orientation where, although they're identical, they still cannot be superimposed. So they can only be symmetrical through a mirror image. We can also see different molecules, macrocyclic molecules, cyclodextrins, amino acids, peptides, oligonucleotides. For the remainder of this talk, we're going to really focus on small molecules. So why is chirality important? These compounds are virtually identical and have virtually identical physicochemical properties. However, they can have very different biological effects. Aspartame has two different isomers. The L isomer, which is a very good sweetener, whereas the D isomer has effectively no flavor. Carvone, the R isomer, has a smell like spearmint, whereas the S isomer has a smell like caraway. When it comes to drugs, though, the drugs can have very different chiral pharmacological effects. The most noted example is thalidomide. The R isomer is a very safe and effective sedative. However, the S isomer is a teratogen and can cause severe birth defects. This is why we now have to do chiral screening as part of FDA approval for drugs. So why do chiral molecules give different pharmacological effects? Shown here in this very simplistic model, we have an active enantiomer interacting with a drug binding site. You notice that all three components match up well with the three components of the binding site, therefore eliciting an effect. It could be an agonist effect, could be an antagonist effect, or an inverse agonist effect. The inactive enantiomer doesn't quite hit all three sites, it only hits the one. So this changes how the drug receptor reacts to that drug. We can also rotate it 270 degrees and get a third different type of binding effect, which could be an entirely different pharmacological effect. This is really important to realize because when you look at today's drug market, 56% of all the NDAs that were submitted in 2020 were referred to as chiral small molecules. So how do you resolve chiral molecules? Well, traditionally this was done through some sort of crystallization, either through chiral salting or derivatization. Today, it's all about chromatography. By and far the largest or most popular way is using some sort of LC technique. But one of the growing areas that we'll talk about today is in SFC, or supercritical fluid chromatography. So how does chiral chromatography work? In this slide, I'm showing what's called the ideal fit model. You have some sort of support with a chiral selector on it. That chiral selector, being solvated, then interacts with a chiral molecule in what's called an ideal fit. That has a stronger bond to its selector, slowing its progress down the stationary phase. You can also have a non-ideal effect, which will have some interaction, but not as good as the ideal effect, again giving different retention times and separation. There are several different types of chiral stationary phases, or CSPs, based on this ideal fit model, where you have a substrate with a selector on it. The main types of these CSPs are either protein-based, cyclodextrin-based, antibiotic-based, brush-type-based, polyacrylamide-based, or polysaccharides. You'll notice, as I'm showing here, the elutability increases as you're going down this line, where the polysaccharides tend to have the largest eluting capacity and makes them the most viable choice for today's pharmaceutical customer. If we actually break that down a little bit further, polysaccharides are based into amylose and cellulose-based polysaccharides. We're going to talk a little bit more about both of these in the next slide. So let's take a look at the polysaccharide CSP chemical structures, starting with either cellulose or amylose, which only differ by their connectivity shown at the alpha and beta positions. We can then do some simple derivatization using isocyanate chemistry to form these phenylcarbamate cellulose and amylose polymers. So once the polysaccharide has been derivatized, it's then put onto a silica gel bead. There's two ways of doing this. One is a coating, where the polymer is taken up into a solvent and dried down onto the silica gel bead, creating what's called a coated CSP. And then there's another process that coats it onto the silica gel bead, and then it is mobilized or polymerized onto that bead, creating what's called an immobilized CSP. So there are some differences. The coated CSPs tend to be a little bit more selective. However, they're also very sensitive to different solvents. Obviously, if methylene chloride is used to dissolve the solvent and put it onto the bead, methylene chloride in your mobile phase would strip the selector off. The immobilized are a little bit different. They're not exactly the same as the coated CSP. A lot of manufacturers will refer to them as complementary. This is because the immobilization process changes the configuration a little bit, giving it slightly different selectivity. The upshot of that is they're very chemically stable to a wide variety of solvents, even DMF and DMSO. So in this slide, I'm showing a couple pictures of both the cellulose and the amylose derivatives. You'll notice that the background creates some very distinctive chiral grooves. This gives steric interference and allows only specific enantiomers to experience or have exposure to the chiral selector. So within that chiral groove, the selector can then interact with the enantiomer of choice through either hydrogen bonding, pi-pi stacking, dipole-dipole stacking, or steric inclusion. Let's take a look at how minor changes in the CSP can have dramatic effects on the resolution of the compound. In this example, prilocaine is exposed to an amylose-based 3,5-dimethyl phenylcarbamate, giving minor resolution. By switching to cellulose, we now have fairly good resolution, but not quite complete. If we then change it from a 3,5-dimethyl to a 3,5-dichloro, we not only get complete resolution, but you'll notice a greater increase in retention time. So far we've looked at things in very simplistic terms. But the truth is there's a lot of molecular interactions going on all at the same time. Some are very strong, some are very weak, some are attractive, some are repulsive, long range, short range. And we haven't even really talked about some of the other effects, like solvent. How does the solvent affect the substrate? How does the solvent affect the selector? How does it affect the selector sample interaction? And then, of course, there's the complexity of the sample itself. Again, more chiral centers, more complexity. Bottom line, there really is no good predictive model that says, based on this structure, this is the certain CSP I should be using. This means there's only really one way of getting resolution, and that is to find the right combination. You have to screen multiple columns under multiple solvent conditions. So the next question is, where do I start? Well, remember, it's still chromatography. So your workflow is just like it would be it for any other chromatography situation. Screen some mobile phases and columns, check the chromatograms for resolution and peak shape, optimize your conditions, recheck your chromatograms, and then you have an established condition. Let's look at a few considerations when setting up your screening. First, let's look at columns. There are multiple CSPs commercially available. So try to incorporate as many as you can into your screening. Consider both coated and immobilized polysaccharides, brush type, proteins, and others. Also consider using CSPs from different manufacturers. Identical CSPs can have different retentive properties depending on the manufacturer's process. Particle size is another consideration. Smaller particles tend to give better resolution, however, at a price of increased pressure on your system. So to get efficient resolution, you want to make sure your system can run the pressures required for those particle sizes. Increasing the length of the column can give you additional retentive properties, however, at a cost of additional runtime. Mobile phases are another consideration. Normal phase, reverse phase, polar organic, SFC, and extended range can all be used depending on the solubility of your sample. However, it should be noted they can give very different results even when used on the same identical CSP. Modifiers can be used to adjust peak shape, and typically are sample dependent. You also need to determine whether you want to run gradient or isocratic. Gradient runs tend to reduce down the number of injections. However, you need to be careful not to miss separations, especially if you're running ballistic gradients. You do also need to re-equilibrate the column in between sample runs. Isocratic tend to require more injections to run, but you're less likely to run tight separations, and you don't need to run equilibration between the samples. So hopefully once you've completed your primary screen, you have at least one or two primary CSP mobile phase hits, but you need to do some optimization. Things to consider: changing your mobile phase composition, maybe augmenting your modifier from one organic base to another, changing the temperature, or increasing the column length. Now, keep in mind that as you're changing these, changing one, it may affect the change in another. So there are different ways of going about this. One way would be use a centralized composite way of augmenting one of these parameters while holding the others constant, or you could do a random walk or a hunt and peck method. And there's also a lot of flow charts that are out there available from different commercial companies and column manufacturers. But once you do the optimization, hopefully you come up with an optimized result. If not, you may have to go back to a secondary CSP and mobile phase hit, and then repeat the process of optimization until you get your optimized resolution. I can't stress this enough. More options give you a higher probability of success. So the more compounds or the more mobile phase CSP combinations that you try, the more likely you are to find success. So we've talked about the need to set up screening paradigms where you're looking at multiple columns, multiple mobile phase combinations. The question really becomes, how do you do this as efficiently as possible? And that's really where it comes down to instrumentation. So let's look at what it really requires to do this efficiently. You need to have a system that automatically switches columns, switches mobile phases. You need to have pumps that will handle pressure for small particle-sized columns. Again, smaller particle-sized columns, smaller columns can be run faster. The faster you run your columns, the more runs you can do, the more columns you can screen. You need to have precise and accurate mobile phase mixing. Again, small changes in mobile phase composition can have a dramatic effect on the resolution of your sample. And then to top it all off, you need to have user-friendly software so you can run all these parameters and analyze the data as efficiently as possible. So with that, I'd like to move into our integrated and modular method scouting systems. So shown here is the integrated I-series, and of course our Nexera system, which is a modular system. So here's the typical method development flow path schematic for one of our systems. Again, having an A and a B pump, looking at 4 different solvent combinations for each, going through 6 different columns here in a single column oven. We can add additional column ovens up to 12, 24, and beyond. We also have different detector options, which will give you possibility of running mass spec, UV, PDA, ELSD. So another option is our dual injection system. It's a unique system that has an A and a B stream that allows samples to be run through different columns in different mobile phases. So people could run samples on an A stream in normal phase and a B stream in extended range or reverse phase or what have you. Again, increasing the capability of running more samples and more different combinations. So up to this point, we've talked a lot about the hardware, the pumps, the ability to use different solvents and different columns and different modes. What we haven't talked about is the software. And software, as you all know, is what really drives the system. So I'm showing here a screenshot of our method scouting system based on our Lab Solutions software. Gives you seamless interaction. The graphics are quite intuitive. So if you want to include or exclude different solvents, columns, change gradients, it's all there. Batch files are automatically created, so if you need to repeat the screens or if you want to set this up for a standard screening procedure, it's all there, done for you. So let's look at some example. Here's one with polar organic screening of flavanone. So we were able to run 8 different columns through a polar organic solvents of methanol, ethanol, acetylnitrile, and isopropanol. And the overlays can show you the very different results for the different columns and different solvents. So here's another example. In this case, we're using 8 different normal phase and extended range solvents in various combinations, running them through 6 different columns and generating different resolutions for 3 different compounds. Another option for screening is our switchable system that allows you to switch between UPLC and SFC. Shown here, we have up to 8 different modifiers running through up to 12 different columns. So using our switchable system, we were able to look at comparative runs between SFC and normal phase or extended range resolution of omeprazole. As shown here, we found various hits and various solvent combinations. So we've seen LC and we've seen SFC screening systems. Here's our SFC screening system. It's a high throughput system that allows you to do multiple samples, up to 24 or more columns, multiple mobile phases. Can also be coupled with mass spec as it's shown here with our 8050 triple quad. Here's an example on our SFC system from a poster that we presented at this year's chirality conference. Devernol was resolved on an IC column using 5 micron. We were able to do some optimization work by increasing the amount of methanol, decreasing the particle size down to 1.67, and increasing the pressure, the flow rate and pressure. This allowed us to reduce the runtime from 4 minutes down to 12 seconds. Here's another example from that same poster session. These 3 analogs of thalidomide, a 6-component mixture, had been previously resolved using an IAU 1.6 micron column. Conditions required a 50% methanol modifier and maxed out at 1.5 mils per minute. On our Nexera system, we were able to reduce the modifier down to 35%, increase the flow rate, and reduce the runtime by 50%. So as a final example, I wanted to highlight this poster from GSK. They have a screening system that has 24 column capabilities with multiple mobile systems in gradient form. This gives them 72 columns and mobile phase combinations per sample, and the method was found to be transferable to PrEP. So in conclusion, I hope that you come away from today's seminar with a little bit better understanding of the importance of chiral molecules and chiral chromatography, the necessity to screen compounds for resolution, and how Shimadzu's I-series and Nexera series of LCs and SFCs with our method scouting software it's a really powerful tool and can really help make that daunting task of doing that screening for chiral chromatography so much easier. So with that, I'd like to thank you for your patience and your time, and I'd be more than happy to answer any questions. I'll turn it back over to Brian. Brian?
Thank you, Steve, and thanks again to our audience. We would encourage you to submit any questions as we transition now to our live question and answer session. Let's again thank Steve for such an engaging presentation. And if you look under the related content list, you can find more information about our analytical solutions and application notes. If you've not yet had the chance, we would ask if you can to fill out the survey questions on the left-hand side to provide us feedback and to request any additional information you may desire. We encourage you and your colleagues to sign up for the Shimadzu e-newsletter by responding to survey question number 3. This monthly newsletter includes information regarding upcoming webinars and events encompassing a wide variety of application areas. So now we're going to start our Q&A session. Again, if you haven't submitted questions through the widget, we ask you to do so. During the presentation, we had a number of requests come in already, wondering whether or not this presentation will be available after the fact, and yes, it will. So for the next 7 days, it will be available through the on-demand link, and after that point in time, it will move to our Shimadzu website, which you can access at www.solutionsforpharma.com. All right, so let's get on to some of these questions that have come in from the audience. And the first one that we have, actually we had a number that were asking kind of a similar question, and it's when you're doing a screening like this, Steve, how many is the minimum number of columns, or how many should I start with when I'm considering doing this type of chiral screening?
That's a great question, Brian. You know, obviously, the more columns you have, the more opportunity there is to find, you know, a combination to give you resolution. However, I think if you have to pare it back, a lot of times due to budgetary issues, you may need to pare it back a bit. I would try to get at least 4 to 6 columns. Try to go more with the immobilized. Because the process allows extended range solvents to be used, will give you more options.
Excellent, excellent. We have a question that's just come in now, and is there a program, I'm assuming a software program, that can identify similarity between molecules for which chiral chromatography systems have been identified for a given molecule and a similar molecule? So is there any sort of predictive modeling that exists right now for chiral stationary phases and molecules?
That's the holy grail of chiral chromatography. There are a number of people that have tried. I know, I believe Chris Welch's group had tried it at one time. Several other people have tried it. To date, I am not aware of anybody that has software out there, commercially available software, that can do any true predictive analysis.
Yeah, I can add to that too, Steve, that I've worked with the Allotrope Foundation, which is focused on creating a data format that can be common among lots of different analytical instruments. And one of their underlying goals is to try and find, through a big data approach, whether or not there can be predictive modeling. But as far as anyone knows, like you said, kind of a holy grail that people are still reaching for. And it's more of a brute force technique still to find that best fit modeling. All right, next question. What are the newest cutting edge chiral stationary phases?
Most of the ones that I've seen out there, especially in the pharmaceutical industry, have come out of companies like Chiral Technologies. They've really pioneered the immobilized CSPs out there. IG, if I remember correctly, was a really big time win for them. The number of compounds that were not found, couldn't find any resolution on anything else, resolved quite nicely on IG. I believe they now have IH and IK out there commercially available. I don't have a lot of information on that, but I do know IG is quite a good one.
Excellent, excellent. We have another question asking about, are there packages to be able to use this type of screening system along with, say, triple quad mass spec detection on the back end?
Short answer is yes, you can use mass spec. I don't know that you need triple quad, but single quad or triple quad would be applicable.
Yeah, and I'll add to that we can use this with any of the mass spec offerings we have up to and including QTOF, depending on what your need is. If you're working in, say, a DMPK space, looking at low-level analytes and things, we can go to that extreme. We can do the discovery, and all of that is still able to be done using the kind of enabling software that was described, the method scouting solution software that allows you to do that quick and easy batch creation. Another question came in regarding, are there current limitations regarding the scale for SFC separations?
Right now, I'm not aware. Most of your scale limitations are due to solubility and loading capacity on CSPs. As far as SFC goes, it, from a preparative standpoint, can be done up to very large scale. So it depends on what scale you're really looking at. If you're getting into production scale, you're probably not running SFC, you're probably running SMB.
Excellent answer. And just to speak to Shimadzu's current lineup, ultimately we have our analytical SFC system that was described in some detail here, and we also have a 150 mil per minute semi-prep SFC. So that's kind of the scale that Shimadzu can cover, is that multi-gram purification up to low kilogram scale, and we have, of course, the analytical complement for that scale up. All right, let me see what other questions.
And you could run columns up to 3 centimeter on that, so that'll give you a lot of preparative capacity.
Excellent. Okay, we have a question asking about acidic modifiers, and will that affect the peak shape of chiral compounds, or is there typically something used in lieu of an acidic modifier when running SFC conditions?
Now, you can use acidic modifiers, or you could use basic modifiers. Typically, if the compound is acidic, you would use an acidic modifier. And conversely, if the compound tends to be basic, you would use a basic modifier. Now, what that modifier can also change not only the resolution, but your peak shape. So if you are running, say, an acidic compound with, and I can't think of an acidic modifier, let's say acetic acid, just for purposes of this question, and maybe you're not getting a good peak shape, or you're seeing a lot of tailing. Switching that up to a different acidic modifier may increase not only the resolution, but sharpen your peaks. Same thing goes with basic modifiers. And then there is also a school of thought of using mixed modifiers. There are some people that swear by using a combination of both an acid and a base, so almost Zwitter ionic, if you will.
Certainly, and for those who aren't used to doing SFC, if you're doing SFC with an alcohol modifier, be it methanol, ethanol, or isopropanol, the conditions will be effectively sub-7 pH. I know typically mixtures of methanol and and CO2 are going to be about pH 5 without the addition of any additional modifier. So, yeah, certainly there are ways to modify those conditions even in SFC in the absence of water. All right, let me see. I think we have one or two more questions. Okay. There was a question that did come in asking, can the same columns be used for reverse phase, normal phase, and or SFC?
Short answer is yes on the polysaccharides. The bigger question is it a wise thing to do? My personal belief is if a column sees any kind of mobile phase conditions with water, I tend to not want to use them in a normal phase or a pure organic phase, just because it seems to me take a long time to drive off all that water to get you back to a completely anhydrous state. But can you do it in a pinch? Yes, you can.
Excellent, excellent. Well, unfortunately, with all of those wonderful questions that have come in, we have run out our allotted time for the webinar today. So I do want to thank everyone for their interesting questions. If we did not have time to answer your question in this forum, we will reach out to each of you individually and answer any of those questions we didn't get to in this session. Once again, thank you all for attending and participating. We will send you an email with a link to view a recorded version of the presentation at any time, and we encourage you and your colleagues to attend our upcoming webinars and the registration link that's located here as well. So have a wonderful day. Thanks again to you, Steve, and we hope to see you in the future at our Shimadzu webinars. Thank you.